study guide. First page for what makes a firefly glow. That is more of a practice to see why protein synthesis matters. So we will talk about that during class, but for this video, we're going to go to the next page to focus on the actual study guide. Okay? And what day is your test? Friday. Okay. What's the purpose of DNA? Which you could say this in a lot of different ways, but it is the instructions of life. Okay, it's a blueprint for making and running an organism. Okay. DNA stand for deoxyribonucleic base acid. DNA molecule is made out of nucleotides. Make sure you know that word. What are the three parts of a nucleotide? Of course, it's the sugar. For DNA, what's the sugar? Deoxyribose. Phosphate group. And that nitrogenous base. Okay. What sugar is found in DNA? I wonder if that's an important question since I asked it twice in the last minute. Deoxyribose. What four bases are found in DNA? A stands for adenine, T stands for thymine, G stands for guanine, and C stands for cytosine. Okay. The base pairing rule is just as simple as A pairs with T and C pairs with G. Thanks, Chargaff, for figuring that out. What bonds hold the bases together? Hydrogen bonds. Sometimes we write that short as H bonds, but make sure you know that the H stands for hydrogen, and we don't want to shorten words on a test. The backbone of DNA, if you cheat and look at this picture, this ribbon part is the backbone, okay? And so we have phosphates and sugar, so phosphate groups and sugar, okay? Now on things where you see the bases here, you need to make sure you really think about what the question's asking. What would the complementary stand of DNA be to the following? So nothing with protein synthesis is going on right now. We are just having DNA base paired together. So A pairs with T, T pairs with A, etc. Okay. Okay. Who discovered the double helix? That was Watson and Crick. And you need to know that they used the work of Rosalind Franklin. Okay. Now, on this picture, I said label the important parts. So hopefully you were able to figure out what all of those were. First, you could call this the backbone. And the two parts of the backbone would be the phosphate and the sugar. I'm not going to write mine out since we just talked about that above, and you know what I'm talking about. This box here contains these three parts, nitrogenous base, phosphate, and sugar. So what are those molecules called that contain those three parts, which DNA and RNA is made out of? A nucleotide. Okay. What else could we label in this picture? You could label all the different A, C, G's, and T's if you wanted to. You will need to know what all those letters stand for, but for this specific picture, I want to make sure you know what these lines are. Those are the bonds that are holding our bases together, which above we said are the hydrogen bonds. In class today, we're going to have a 3D model of DNA that you can look at, but I just want to show you this one on the video, and let's name all of our parts that we need to, okay? We have our backbone, which is going to be made out of phosphates and sugars, which we can't really see here. They just have the ribbon. That's what the backbone is made out of. Then we have our bases in the middle, and they're paired using hydrogen bonds. Okay. Okay, next page. What is a gene? A gene is going to be a region of DNA which codes for a trait. And what controls a trait is going to be the proteins. Okay. We think of genes as controlling a trait, but using the word protein is a more exact definition because, as we're going to learn in the following weeks, there are a lot of traits in our bodies that are controlled by multiple genes. So, but for right now, we just need to think of one segment of DNA that we're going to do protein synthesis with. Okay, and that's a gene. All right, if we put these in order, we would start with DNA because that is like the master copy of all of our instructions. Then I'd go to RNA. Those are smaller pieces of those instructions. RNA is used to make proteins. And our proteins determine our traits. And this idea of DNA, RNA, and proteins is called the central dogma of biology. That's how important this process is to all the different things that we study in biology. Okay. RNA stands for, if you know what DNA stands for, this one's easy. Ribonucleic acid. Okay. What molecules are both DNA and RNA made of? Nucleotides. What are the three differences between DNA and RNA? Okay, first, we have different sugars. And I'm going to draw like a little table here. So DNA will be here, and RNA will be here. DNA, RNA. What sugar does DNA have? Deoxyribose. And RNA has ribose. Okay. Next, we could look at the bases. What do they have in common? They all have adenine, cytosine, and guanine, but DNA is going to have thymine, where RNA is going to have uracil. Okay, and the third important difference is going to be that DNA is double-stranded and RNA is single-stranded. Okay, we could talk about other similarities and differences between DNA and RNA, but these are the major ones that you definitely need to know. All right, three tops in RNA and what are their jobs? For looking at our three different types of RNA, I'm going to go to the amoeba sisters because they have nice visuals here, and then we'll finish filling out the sheet. Loading. Well, I don't think it wants to give me video as we are recording. Time to problem shoot. Okay, let's try it again.
It's going to help deliver the message. There are actually 3 types of RNA, but don't.
Okay, well, let's just go ahead and talk about it. I will show you videos and visuals in class because I think it's good to see what these are doing as we're describing them. Okay, so mRNA, messenger RNA, carries messages from the DNA to the ribosome. tRNA is the transfer RNA. It has that amino acid hat that it carries to the ribosome. And remember, it also has those anticodons that match up to the mRNA's codons, so it knows which amino acid to bring. Then we have the ribosomal RNA, which makes up the ribosome, which is where protein synthesis occurs. Okay. Protein synthesis can be broken into 2 parts. Part 1 is transcription, part 2 is translation. And transcription, we could answer this with a long answer, but I'm going to tell you what I normally, a shortened but still has all the important points version, is we go from DNA to messenger RNA. Okay? For translation, we can do similar. The messenger RNA gets coded in order to make what? What are we making in translation? We're making a protein. Okay, you could also call this an amino acid chain. Okay, all right. Given the following sequence of DNA, transcribe and translate it into a protein. So if we look first to transcription, step 1, transcription, okay? We're going to base pair, but remember, unlike DNA to DNA, we need to use RNA, so we're going to have uracil instead of thymine. Okay. All right, transcription is done. Bam. That one is the easy part. Next is translation. So it says the Google codon chart. So, all right, going to Google. If it'll load. Okay, I'm pausing it as it loads. Well, we're conveniently having a problem with the Wi-Fi at this moment. So this is a little fuzzy, but it should work, and you should be able to find a better one anyway. So if we look at our first codon, and I like to go ahead and split these into 3s. It's easy to keep track that way. Okay, we have AUG. So on your chart, you start from the middle and work your way out. So A. No. A. U, G, and we know that's methionine, and you need to remember your rules for translation. Okay, we cannot start until we have methionine. So if this was anything other than methionine, you would just have the codon here and ignore, don't put this, cross that out, until you see a methionine, then you would begin your translation. So remember translation, we're going from an mRNA to a protein. Okay, so GCC, go back to your chart, GCC, alanine, so that should be ALA. Okay, I wrote ALA here. And you go on until when? Until you get a stop codon or until you see the end of whatever you're given, because maybe it's just a fragment of a gene that people want you to translate. Whoa, did you catch the mistake Mrs. Rhodes made? Yep, put a T instead of a U, rookie mistake on a video for everybody to see. But if you are translating and you see a T, you could change it, because that's a great time to catch your mistakes, because obviously there are no T's on your codon chart. Ooh. Okay, U, G, G. Okay, this one was nice and not so tricky because it starts with a start codon and ends with a stop codon. But don't forget those rules, because you might see something on the test that could trip you up. Okay, so transcription is going to occur. That's when we go, let's think about what transcription is. DNA to RNA, or messenger RNA. That's going to occur in the nucleus. That's the whole reason we have RNA, is because the DNA cannot leave the nucleus. Anyways, that's one reason we have RNA. Where does translation occur? If we look at our picture here, this would be the nucleus. So out here would definitely be in the cytoplasm. And where does translation actually have to occur? On this blob, which is called a ribosome. So our answer here, where does translation occur? I would put ribosome. You could say the ribosome within the cytoplasm, but we know the location is accurate to this picture. It is outside of the nucleus at a new location at the ribosome, which is in the cytoplasm. What's a codon? We're going to say 3 bases of where a codon's found. What type of RNA? Messenger RNA. And they code for a and amino acid. Remember, if you look back at our chart, a codon, such as GAU, codes for an amino acid. What is a protein made up of? What are the building blocks of protein? That would be amino acids. And that's what we're building with translation and protein synthesis. So let's look at our picture here. We have two different processes going on. The first one here in the nucleus would be transcription. Okay, that's where we have DNA, double-stranded, so we know it's DNA here. What's being made from the DNA? It's going to be your messenger RNA. Okay. Messenger RNA, and that's all that transcription is, and it happens within the nucleus. Al on Z.